Welcome to Switzerland. Did you know that our country is not just a great place to visit, but also your first-class business location? We are centrally located in Europe and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Welche dir? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic, and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
For us in the Aditya Birla Group, Odisha has been one of our most preferred investment destinations. Chief Minister Shri Naveen Patnaik's exceptional leadership has been the singular factor in the rapid progress of Odisha. The state government has leveraged its competitive advantages, including its progressive policies and governance framework, are a big pull factor.
I welcome you to Odisha. Come, join the Juggernaut.
In healthcare, a vast ecosystem is helping therapy innovators become more patient-centric and more effective than ever before. Cytiva supports researchers, biopharma, and clinicians in the pursuit of more targeted treatments. In discovery, speed and accuracy are everything. The faster and smarter researchers can work, the more patients benefit. To ensure that new, more targeted therapies become reality, we're equipping biomanufacturers with the tools and expertise they need to produce cost-effectively and quickly. How can tens of thousands of individualized patient doses be manufactured quickly, efficiently, and securely? The answer, with a healthy dose of expertise and innovation. We're creating processes and solutions that are scalable and address complex needs. We work to deliver diagnostic tools and other technologies to bring the right therapy to the right patient at the right time, so that life-saving medicines come within the reach of more people and could save more lives. Cell culture, protein purification, biomarker imaging, and analysis. It may not be what most think about when envisioning the future of healthcare, but this is what we do. Our part in changing the world. Advancing and accelerating. Discovery, manufacturing, diagnostics, future therapeutics.
road to regulatory compliance is long and filled with choices. With a combination of standards, process, and service, USP can help guide your path to compliant, quality products. Trust the standard that sets the benchmark for medicines. Our robust and collaborative scientific process is part of our comprehensive approach. We offer reference standards that are tied to USP monographs to help you minimize risk and enhance your confidence in compliance. Reduce time and resources spent developing in-house standards and streamline your path to regulatory compliance. We work with independent, volunteer scientific experts who rigorously review and approve our standards, which undergo testing by USP and other laboratories around the world. Ongoing testing of our standards helps ensure the quality of your product over time. And only USP reference standards are linked to official USP NF monographs that provide specifications for the identity, purity, and potency to meet FDA requirements without further in-house qualification and the needs of our global customers. With USP, you have access to our in-house scientific experts to guide you every step of the way. In addition to our best-in-class USP reference standards, we offer training, education, and other services. Our online resources help smooth your path to regulatory compliance. Our standards provide precise testing and validation guidelines, as well as reference samples for testing. Drugs can be made consistently every time. We are more than reference standards. We provide a unique combination of standards, process, and service. From buying to applying standards, we support your bottom line with reference standards that have been rated best in class by our customers. Talk with us to find out how we can help you navigate your journey to regulatory compliance. Contact USP at usp.org.
Welcome to Switzerland. Did you know that our country...
Department of Biotechnology, Ministry of Science and Technology, Government of India and its public sector undertaking Biotechnology Industry Research Assistance Council, BIRAC, are organizing the second edition of Global Bio India from 1st to 3rd March 2021 on digital platform. The first edition in 2019 at AeroCity, New Delhi was a huge success with participation from over 25 plus countries, 190 exhibitors, 2,500 plus delegates, 300 plus startups, 50 plus incubators, 60 plus research institutes, 800 plus business meetings scheduling and representation from 9 states. India offers a unique destination as innovation hub and biomanufacturing hub for the world. Large talent pool, growing capacities for frugal innovation and manufacturing of affordable, globally competent products and technologies in healthcare, agriculture and allied area, food and nutrition, waste to value, sanitation and clean energy solutions. biotechnology sector is recognized as the sunrise sector that would have multiplier cascading impact on achieving India's USD 5 trillion economy target by 2024. Government of India initiatives reiterates the significance of biotechnology sector as the sunshine sector. Global Bio India 2021, the largest bio event, would showcase India's biotech sector strength and opportunities at global level. Global Bio India 2021 is expected to draw around 5,000 plus delegates from across the globe. Department of Biotechnology, BIRAC, along with its autonomous institutes, has been at the forefront in the nation's fight against COVID-19. The tableau, India Fights COVID, by Department of Biotechnology, Ministry of Science and Technology, showcasing significant contribution of biotechnology sector in the development of vaccine, under Make in India mission, India is now gearing up to become innovation hub and biomanufacturing hub for India and for the world. This happens to be the most exciting time to host Global Bio India 2021, India's largest biotech event. Led by DBT and BIRAC in partnership with CII, ABLE and Invest India. Together, we can transform billions of lives worldwide. Join us on the biotech journey. Global Bio India 2021. Jai Hind.
Welcome to Switzerland. Did you know that our country is not just a great place to visit, but also your first-class business location? We are centrally located in Europe, and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Decidia? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic, and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
For us in the Aditya Birla Group, Odisha has been one of our most preferred investment destinations. Chief Minister Shri Naveen Patnaik's exceptional leadership has been the singular factor in the rapid progress of Odisha. The state government has leveraged its competitive advantages, including its progressive policies and governance framework, are a big pull factor.
I welcome you to Odisha. Come, join the Juggernaut.
In healthcare, a vast ecosystem is helping therapy innovators become more patient-centric and more effective than ever before. Cytiva supports researchers, biopharma, and clinicians in the pursuit of more targeted treatments. In discovery, speed and accuracy are everything. The faster and smarter researchers can work, the more patients benefit. To ensure that new, more targeted therapies become reality, we're equipping biomanufacturers with the tools and expertise they need to produce cost-effectively and quickly. How can tens of thousands of individualized patient doses be manufactured quickly, efficiently, and securely? The answer.
With everyone. Thank you so much. Sai, over to you.
Yeah, so directly I will be opening or?
Right, you will be opening.
Okay.
Yes, ma'am.
Dr. Sai, we are going live soon.
Okay, please.
We will start at 6 o'clock exactly, isn't it?
Yes,
Now.
please. You can go live.
Are we live now?
Sir, we are live now.
Are we starting then?
Yes, ma'am. We can start the session now.
Yes. Very good evening to all. I am Dr. Suchita Ninave, and on behalf of Department of Biotechnology, I welcome esteemed panel members and August gathering to this exciting and very important session for understanding the technologies that would help us to address the needs of tomorrow. Biological science is expanding its knowledge frontiers speedily. The progressing insights into biological processes offer a broadening array of options to develop incremental and differential innovations across the medical, agricultural, and industrial biotechnology sectors. In pursuit to anticipated demands of emerging technologies, Department of Biotechnology is supporting programs in various emerging areas like genome editing technologies, nanobiotechnology, artificial intelligence, system biology, alternate medicine and targeted therapeutics, stem cell research, etc. Here in this session, we will have an opportunity to listen a lead talk by Professor Melanie Wilhelm, Executive Chair of the Biotechnology and Biological Sciences Research Council of UKRI on engineering biology. Thank you, Professor Melanie, for your presence today. Further, we will have talks by eminent experts on alternate medicine, precision biology, stem cell modeling, and clean energy. It is expected that this session will provide some understanding of integration of evolving manufacturing and analytical technologies to address user requirements of development of robust bioprocesses at reduced cost and increased efficiencies. With this, I request Dr. Prabhuda Kundu, co-founder and the managing director at Premas Biotech, to present his introductory note and set the tone for this session. He himself is a biomedical engineer and has interest in protein engineering, bioprocess development, lean transformation, and other emerging technologies. He is a perfect moderator for this session, I can say this. Now over to you, Dr. Prabhuda.
Thank you so much. Thank you. Good evening, good afternoon, and good morning to everyone who has logged into this very exciting panel discussions. At the onset, I'm very grateful to the DBT Secretary, Dr. Renu Sarup, who initially contacted us and said, you know, if I'd be interested, Dr. Manish Dewan, and Dr. Shaishyam for his tireless follow-up, and the organizers for putting this very important topic for discussions, emerging technologies. Thank you so much, Dr. Sucheta, for your opening remarks. We have a very distinguished and illustrious panel for the session today, and I will spend a few minutes introducing the topic for the panel, Emerging Technologies. However, I will go over the last 12 months where we have seen the world has been thrown into a turmoil of gigantic proportions. Jobs, livelihood, and general well-being, emotional health has been turned upside down. Both governments and humanity has struggled with it. However, the pandemic is ebbing and flowing across the globe, but doesn't seem to be going anywhere. Despite this, a fantastic, collective, and focused global effort has been working to counter the COVID pandemic, where the global scientific force has played a major role. New vaccines, new therapies, new ways of collaborating, and new ways of working have been established. COVID-19 has forced communities and countries to reassess and understand the importance of education of science and scientific work. Today, ACE2 and RBD are common language, and neutralizing antibodies are being sought after, and so are new therapies. With that, there is a recognition for new technologies and the impact it has brought, whether it's vaccines or therapeutics or medical technologies. New modalities are the prime example of such innovation that there is on display for all of us to see. With this, it brings me to our panelists. Each of our esteemed panelists are working or aligning themselves with new technology, which is in the future will play a very critical role. They are in various stages of development and in varied fields. But one thing is certain, it is going to be a very interesting and exciting talk. They will cover engineering biology by Professor Melanie, to oral delivery of therapeutic proteins, to the future of foods and milk, to highly precise analytical technologies and techniques, to understanding of pathways for new tools of cancer therapies, and from wealth, which is there in waste. With that, I would like to invite Professor Melanie Wellum to begin the session as the lead speaker today. A brief introduction, if I may, for Professor Wellum. Professor Melanie Wellum is the Executive Chair of Biotechnology and Biological Sciences Research Council, UK, and Innovation, UKRI. Prior to this, Professor Wellum worked at the University of Bath as the Professor of Molecular Signaling. She holds a degree of biochemistry from the Imperial College London before undertaking a PhD at UCL. Over to you, Professor Melanie.
Thank you. And it's a real pleasure to be here. My laptop, although we tested the sharing the screen, there we go, it's now starting to share. So hopefully you can all see the slides there. So it's a real pleasure to be here, and I'd like to thank Dr. Sucheta and Dr. Prabhada for very kind introductions. The organisations I work as part of, UK Research and Innovation, supports partnership across researchers in academia and industry, not just in the UK, but globally as well. And we have a long and strengthening partnership supported by our international offices around the globe, and particularly with the UKRI India office. We work closely with a number of Indian government departments, particularly the Department for Biotechnology, and we've co-invested in a whole range of programs. So this is a really significant partnership, and really it makes being here with you today sort of very special to help celebrate that and think about how we can go forward. So in terms of what we do in BBSRC, which is the organisation that I lead, our vision, focused around biotechnology and biology, is really about how we push back the frontiers of knowledge and how we can then use that knowledge to create a healthy, prosperous and sustainable future. And I think that's really what we're talking about here. What is the potential of some of those exciting biologically based emerging technologies to help us think about building back better after the pandemic, a greener, more resilient future? I think BBSRC has had a tradition of horizon scanning, seeking out those emerging technologies that we need to be considering, we need to be investing in, we need to be building capability to equip our researchers and our businesses for the future. And we invested over 20 years ago in genomics capability and applications. We invested in systems biology, and approximately 10 years ago or so, we started to identify synthetic biology as a new emerging technology that we wanted to be growing the capability for in the UK. Now, synthetic biology in conceptual terms can really be considered as applying engineering principles to biology, thinking about designing, redesigning, and creating biological devices, systems, components, and processes. And another way to think about that is actually how you can harness the tremendous power of nature, the power of biology, and turn that and apply that to improving lives. So it's sort of nature or biologically inspired work. We've been busy delivering a roadmap that was set out in 2012, and we've built capabilities all across the country, as illustrated on this map here, which have different focus on different aspects of synthetic biology and in different systems. And in a moment, I'll just be elaborating a few examples of the outcomes of some of the research that those centres that have established that capability have actually generated. So you might then be thinking, why am I talking about engineering biology? Well, always with a view on the horizon about what's coming next. We recognise that synthetic biology is tremendously powerful at creating new bio-based processes and systems through the design, build, test, analyse cycle that really is at the core of synthetic biology approaches. But what we need to do is to then realise the benefits of the knowledge that's being created and discovered. And to do that, we sort of need to look much more broadly about how you draw out and draw on those discoveries, translate them, commercialise and scale up. So engineering biology really is about realising the potential that synthetic biology offers, but sort of encapsulate some of the wider ecosystem and the capabilities that you need to really support exploitation of that knowledge, to deliver societal benefit, economic benefit. And, you know, there's a huge, this is the topic of your conference, there's a huge market out there around the bioeconomy, and there are real opportunities for the future for transformation. And some of those opportunities are kind of illustrated on this slide in terms of some of the themes where applying engineering biology principles can actually has the potential to have quite transformative impact. So whether that's in biomedicine, in clean growth, in food systems, or in environmental solutions, there's a whole range of different application areas. And of course, discovery research continues to be really important and underpins all of those themes. We need to continue to think about bio-inspired design, bioengineered systems and cells, and novel materials. What I want to do in my next few slides is to highlight some of the outcomes from the investments that we have made as part of UKRI in building that synthetic biology capability, and where the opportunities sort of illustrate how that can take us into the future with opportunities. So my first example sort of relates to the broad area of biomedicine, and this is research that's being led by the Bristol Synthetic Biology Centre. They've developed a new technique, which they called Zentide, whereby they've used synthetic biology to engineer bacteria to generate peptides, so to build peptides within the bacteria. This is very different to the chemical synthesis of peptides, which is normally used. It has much lower economic and environmental cost, so clear benefits there already. The researchers in Bristol set up a spin-out company that BBSRC, my organisation, helped to support, and they're taking inspiration from nature in developing novel adhesive peptides. So they look to the marine mussels that cling to intertidal rocks for their inspiration. And they're developing these adhesive peptides for medical and defence applications. So really interesting area. Next example sort of relates to that broader clean growth, the greener future. And this is a spin-out company, Colorifics, from the Open Plant Synthetic Biology Centre, which is a collaboration between the University of Cambridge and the John Innes Centre at Norwich. This is the first company which is using biological processes to produce, develop, deposit and fix pigments onto textiles. Again, inspired by nature, which, you know, we are filled with wonderful colours around us. So inspired by nature, recreating those biosynthetic pathways in microorganisms to generate different pigments is really at the core of what Colorifics are actually doing. And they've demonstrated that their process uses 10 times less water than traditional dyeing methods, doesn't require non-renewable petrochemicals, so has broader environmental benefits. And what's really interesting is that they use the microorganisms directly to transfer colour onto fabric. So it's a really interesting model, and as I said, is one of the first companies to actually take this type of approach. In the food space, Deep Branch Biotechnology, which is a spin-out from the Nottingham Synthetic Biology Centre, has brought together a consortium of partners across academia and business to tackle this problem of transforming waste, in this case carbon dioxide, into animal feed, into protein. So they're using they're capturing CO2 from the Drax power plant in the UK, which is a biomass power plant. They're capturing that CO2, they're using it as a feedstock for their microbial fermentation, and the microbes in that fermenter have been engineered to produce protein, which will then be harvested and used for livestock feed for applications, I think initially poultry and aquaculture feed. They've actually proposed that this process has a 75% smaller carbon footprint than traditional methods of producing animal feed, and clearly reduces pressure on our food production systems by providing alternative sources of protein, and more than that, actually utilising waste CO2. And we all know how CO2 is contributing to the changing climate and how important it is to reduce CO2 levels. So again, a really interesting project which touches on many of those different sectors and opportunities I talked about. Building on sort of environmental solutions, C3 Biotech, which is a spin-out from the Manchester Institute of Biotechnology, are looking at using engineering biology approaches to biomanufacture hydrocarbon fuels from using major industrial waste sources. And they've got projects where they're looking at production of bioethanol, at precursors of aviation fuel. But interestingly, they've also got a collaborative project supported through the Newton Bhabha Fund with Indian collaborators to look at creating high-value chemicals using waste from paper mills as their feedstock. And again, really interesting, and I think we'll hear more later in this session about wealth from waste. So this is an example of one of those collaborations that we're really, really happy to see and to support across the UK and India. My final example is something which is really rather broader, and it's taking transformative and emerging technologies from other sectors, in this case artificial intelligence, and applying those to synthetic and engineering biology. This example is a spin-out company, Lab Genius, which was set up by a group of PhD students from Imperial College in London a number of years ago. And they have really pioneered the use of artificial intelligence algorithms to predict genetic changes which will improve biological function and so feed into biological design in synthetic and engineering biology. This is broad. It could be applied in areas of sensors, in areas of materials, in areas of therapeutics. And that latter area is really where they focus, and they've been really successful in raising funding to support their work looking at protein therapeutics using their novel AI approaches. But clearly the application could be much, much broader. So I hope in that very quick presentation, I will have demonstrated how, there are many, many biotechnology solutions there inspired by nature. And I think we can look forward to how the application of synthetic and engineering biology can really help us with that transformation and deliver a sustainable bioeconomy in years to come. Thank you very much for your attention.
Thank you so much, Professor Melanie. That was a fascinating talk. Thank you so much. Really very grateful to have you. And I think we will take all the questions and all at the end. So now moving on to Mr. Nadav Kidron. Mr. Kidron is the CEO and president of Oramed Pharmaceuticals, Inc., and he will be talking about ingesting versus injecting, the future of administering therapies. Mr. Nadav Kidron serves as the CEO and director of Oramed Pharmaceuticals, which he co-founded in 2006. Mr. Kidron is an entrepreneur whose experience includes senior executive roles in a wide range of industries. He co-founded Entera Bio as a joint venture, founded Oramed, as well as DNA Biomedical Solutions. He's a member of the IIT board. He holds a bachelor's degree in law and an international master's in business administration. So I take this opportunity to welcome Nadav to the panel. Over to you, Nadav.
Thank you, Prabhuda. It's a pleasure to be here, and thank you for inviting us to be part of this exciting conference. What I'll try to do over the next few minutes is to really share with you a concept that I think fits very well into this whole innovation panel. Because if you think about it, a few years ago, if you went to medical school, you will learn that part of biology is that peptides cannot be administered orally. That was the notion. You need someone to administer it to everyone who needs to get the vaccine. Imagine you can walk to the pharmacy next door, you give your credit card, you pay the money, you take the vaccine, you go home, take a bottle of water, you swallow it and you're done. That is a game changer. So if you think about vaccine that you need to take every 6 months or once a year, and you take away all the logistics, so it's way beyond the patient's compliance. So when we talk about the concept of delivering peptides orally, it's way beyond the obvious. It's easier to administer. And I would love to share with you a few of our slides and share about the pipeline of what it is that we do in order for us to really understand that there is something very exciting going on here. And let me just show the screen. I hope this time it's going to be going better than beforehand. Okay, I just want to say one more thing and then I'll share that. There is one major difference when we deliver something orally versus injection. And this thing that I'm going to say now is the key for everything we're going to discuss. And that has to do with the following. When we give someone an injection, then the API goes directly into the bloodstream. When we give something orally, then it actually goes into the liver as the first pass. And going into the liver as the first pass can be a huge advantage in many, many of the cases that we give those peptides. So allow me to share with you a few of the things that will explain and demonstrate why we believe that this is going to be one of the most exciting things for us to embrace. So I hope you can see the screen. And I want to start with a little bit of the obvious, or maybe not so obvious. How do we do it? How can we take a peptide and deliver it orally? So in order to explain that, we need to understand what prevents us from doing so. And what prevents us from doing so is obviously the degradation and the size of the peptide. By developing a protease inhibitor and putting it with an enhancer in a softgel capsule, we're actually able to deliver the peptides, the API, intact into the intestine. And basically, because of the change in the pH, the coating dissolves, we give it the protection, and with the enhancer, we get it into the portal vein, and from there it goes into the liver. So let's see what are we working on at Oramed. So oral insulin is our flagship product. And oral insulin for type 2 is undergoing now a phase 3 under the FDA. And next year, we're going to have results of a phase 3, 100 years since the discovery of insulin. When insulin was discovered, the first way they tried to administer it was orally, and it failed. 100 years later, finally, the technology is around. 100 years later, we're going to see the results of giving insulin orally. Now, I'm going to share with you a secret that we probably know what the results would look like because we've already given it to thousands and thousands of doses, and we saw that oral insulin is safe, oral insulin works, and there's basically no side effects because of the oral delivery. So when we look into the future, and I just heard a lecture last night that in the United States, when you talk about people over the age of 50, we talk about 50% that are overweight. There's one thing for sure that we're going to see in the future, and that's a lot of diabetes. When we look at the graphs of how many diabetics there are, you see the sharp curve, and we know that with our lifestyle, with the consumption of calories, and unfortunately the lack of exercise, only one thing is for sure, a lot of diabetes. Oral insulin will enable us to treat diabetes in a superior way. Why? Because of the following thing. And this is, I think, the key thing for everything we're discussing. The liver is the organ that regulates the secretion of the glucose and the insulin into the bloodstream. How does the liver know what to do, how to do it? Through the signaling. So most of the type 2 patients have a huge problem that there's an excessive production of glucose during the night. By giving them oral insulin at bedtime, that signals to the liver, basically telling the liver, Mr. Liver, you've got too much sugar here. The liver shuts off the excessive production of glucose. Those patients wake up with a lower level of fasting blood glucose, and therefore the A1C is going down in the most elegant way, dealing with the source of the problem and not with the effect of the problem. Not just giving the Tylenol, but unrooting the issue. That's why we believe that oral insulin is the biggest game changer to the world of diabetes after 100 years of since the day that we discovered the insulin. I could go on and on and on, but the idea is really to give a snapshot of what it is that we do. So I will just stop here and I will welcome you, anybody who wants to be part and to see the innovation that's going on in the world of diabetes, in the world of oral delivery of peptides. Please join us, and we'd love to answer questions down the line. So thank you again for the opportunity, and looking forward to hearing the rest of the speakers here.
Thank you so much, Nadav. That was a fantastic view into the pipeline that you have and the technology. I'm sure there'll be lots of questions that Dr. Sharsham would be having and we would be getting. And we'll try and collate it to you, and please be with us, and I think we'll have the question answers towards the end. If that's okay, Nadav. Thank you so much. And I must thank both the speakers for being fantastic on time. Actually, we are 1 minute ahead of time, so that's fantastic. So now we have Dr. Ori Kohavi, who is the co-founder and CTO of Remilk. He's going to be talking on precision fermentation-based dairy products. Dr. Ori Kohavi is the co-founder and Chief Technology Officer of Remilk. He is a PhD and postdoctoral from the Weizmann Institute of Science in the area of protein surface interactions and worked as the head of discovery in Mitoconix Bio Limited. His experience is in the area of protein chemistry production and quality control. With that, Ori, welcome, a very warm welcome to you. And I think we are all very excited to hear, at least a couple of panel members did say, oh, we are very happy that Remilk is on the panels. Please, over to you.
Thank you very much, Prabuddha. It's a pleasure to be here, and thank you. I want to thank the organizers for inviting me. So I will talk, okay, I'll share the screen. I hope you can see. Yeah, so I will give a brief overview over about our perspective on precision fermentation-based dairy. and obviously it's going to be a bit of a high level, but this is the time we have. So at Milk Festival, we believe that we are currently on the verge of a very deep disruption in the way food is being produced. We see that the current model of industrial livestock production is a very inefficient way in supplying the world with food, very volatile and low margin. So we're mostly talking about economics as the key driver to this change that we think we are about to see in the next few years. This is true for, of course, the entire food industry, but it is, I think, very much true for the dairy, the current dairy industry. And we can see that the market is definitely changing rapidly, and this whole rise of the flexitarian movement, we have 40% of the Americans currently define themselves as being flexitarians. These numbers are even higher in many parts of Europe. And the traditional dairy industry, which is based on animals, of course, is already balancing on a knife's edge, and it is trying to live up to the expectation of this flexitarian movement. It has a very modest growth compared to plant-based dairy, which is rising very rapidly. So our mission is to actually replace the current, basically replacing the cow, if you will. And we believe that the way to do it is use the precision fermentation. This is already, of course, a proven technology that did marvelous work ever since the 1980s in disrupting and changing current animal-based products. And we can see that the products that are being produced using precision fermentation are superior in every conceivable way over the former animal-based products. Of course, we have the examples of non-animal insulin. Ever since it was being produced recombinantly, it very quickly replaced the animal-based insulin. We can see it also in other types of products. One of them is the chymosin, the cheese-making enzyme, which also replaced pretty much, most, not all, but most of the traditional way of producing chymosin, which was animal-based. So once you have this product which appear and they're of course superior and more economically viable than animal-based products, they quickly replace them. And we believe the same is going to happen in the dairy industry very soon. And one of the nice things regarding the dairy industry is that by producing only about 3% of the milk bottle, which is actually the most functional part, the proteins, you can actually disrupt this industry, which is even in the food industry, it's the most economically vulnerable part. And talking about milk as a material, it is pretty clear that milk is very functional. It allows you to form very desirable products that are consumed all over the world, such as cheese. And basically, the functional part of as I said, of the milk is the protein part. We're talking about mostly the caseins, but also whey proteins. And when we address the non-dairy alternative, which are as of today mostly based on plant proteins, they do a fairly good job in replacing liquid milk, and are pretty successful. But once you reach more complex products such as cheese, you can see that there is still a pretty big gap in what these products can actually do. So they don't do a very good job in replacing, especially I would say cheese. product. So in order to actually bridge this gap, you need to have the proteins which are actually identical to the animal-based dairy protein. And this is exactly what we do at Remilk. So of course, very briefly, we have the DNA of the different milk proteins. So in Remilk, we work on all the major milk proteins. And then we transform different types of microorganisms that become producers of these proteins. And by using fermentation followed by downstream process purification and formulation, we can actually achieve identical proteins, identical structure to the ones that are present in milk, and they can function in a way that is identical to the natural animal-based proteins. The key, I think, to this and the holy grail is what's called the casein micelle. It's basically an aggregated structure of thousands of different individual casein molecules. And this is the main, I would say, the sole responsible for the different attributes, the good qualities of what we expect from dairy products, the ability to melt and stretch and have all these nice functionalities, all come or mostly comes from this casein micelle. And we were able at Remilk to actually use individual caseins and using a very simple and patented approach to actually construct casein micelles, which are identical in every kind of test that you can imagine. And they are functional. They're able to actually coagulate using the exact same processes that are used at the dairy industries, exact same enzymes and cultures, and to actually reach identical product. Taking this micelle, you can again form cheese, which proved to be indistinguishable from regular dairy cheese, and have all these qualities in terms of flavor, smoothness, softness, stretchiness, etc. It has no cholesterol in it. It has no lactose in it, and it was again created using the same industrial cheese making process. And just to end with this nice slide, that this shows that, I mean, this industry, why this industry is going to replace, the traditional animal-based industries. So it's simply much more efficient in every way you can think of. Yeah, I'll be happy to address some questions later on.
Thank you, Ori. That was fascinating talk. I'm sure that's got few people thinking already of the opportunities. And thank you so much. Very, very nice. As we move on, we have Mr. Varun Deshpande, who's the Managing Director of the Good Food Institute in India. Varun serves as the Managing Director of the Good Food Institute, and where he's focused on building the future of food by working with scientists, foundations, government, entrepreneurs, and corporations to build the alternative protein sector across the country. He spent several formative years studying chemical and biomedical engineering at the Technology Hub of Carnegie Mellon University. He went then to work on implementing digital health in India and in the United States, helping a lot of vulnerable populations and trying to make a systemic approach to healthcare. Varun is a leader in the Effective Altruism movement, which seeks to investigate and target the world's most pressing problems. He's been a leading young voice, and he's also been voted as India's 25 most influential young citizens by GQ. Varun, over to you. Delighted to have you here. Please.
Thank you so much. Thanks, everyone, and thank you to the organizing committee for having me here. Actually, this is pretty convenient for me to go right after Ori because we're speaking about somewhat similar topics. But I'll jump right into it. Could you confirm that you see this presentation?
Absolutely.
Okay, great. So as Prabuddha said, thank you so much for the introduction. My name is Varun Deshpande. I'm Managing Director at the Good Food Institute India, and I'm here to talk to you today about smart protein or alternative protein, which is we see as a key pillar of India's emerging bioeconomy for the next decade. Just a really quick introduction about us. So we are the Good Food Institute India. We're part of a global network of nonprofits spread across India, East Asia, Singapore, Israel, Brazil, Europe, and the US. And we work across business, science, and policy to advance and establish the alternative protein sector or smart protein sector, which in India means building it from the ground up. If we look at the major question that guides or underpins our work, and this is very similar for many people on the panel here today, and it's something that Ori just alluded to before this as well, we're focused on figuring out how we feed 10 billion people on this planet by the year 2050, 1/6 of whom will be in India, through systems which do not continue to place tremendous stress on biodiversity, scarce natural resources, a whole swath of problems under planetary health, as well as public health. And then when you analyze this challenge, one of the things that seems to come up over and over is demand for animal protein. And it becomes pretty clear that we need a diversification of protein production platforms for human food applications. So if you look at, for example, the chicken, which is the most efficient animal that is farmed for meat, the chicken takes in 9 calories of input for every 1 calorie of output in the form of flesh. And this is, in highly optimized operations that cannot really get much better over time. And it's not just the efficiency argument. There are also a number of sustainability issues with animal factory farming, as it's called. I mean, you can look this up. I won't spend a lot of time on this, but essentially the United Nations, the Intergovernmental Panel on Climate Change, the Eat Lancet report, the Food and Land Use Coalition, I mean, a whole slew of organizations and governments have already identified that if we care about ocean dead zones, ocean acidification, greenhouse gas emissions, deforestation, species loss, pretty much any problem under a climate and sustainability umbrella, then we should care about our protein production platforms today. As an example, industrialized animal agriculture or factory farming, our current protein production platform, is responsible for more climate change than all emissions from the entire transportation sector combined. And then, of course, as Prabuddha said at the beginning of this, we are having this conversation at either the middle of or the end of a global pandemic, an unprecedented situation, wherein we're having to have this conversation remotely, right? And so it's worth noting that COVID-19 is shining a light on a lot of these systems in ways that have not been done before. And if you look at any of the causes of major public health risks, whether zoonotic diseases, which have been identified as the major source of the next pandemics, as they have been for pandemics in the past, as well as antimicrobial resistance, we really have to examine the way that we're producing protein using all of these animals, because 70 to 80% of all antimicrobials consumed globally are actually fed to farm animals and not to humans. Worth noting also, just to underline, that this diversification of protein supply is particularly important in low and middle income countries like India, because the resilience of food production is very, very important in these countries, both in terms of safety as well as in terms of economic systems. So we've just had, at the beginning of this year, yet another major avian flu outbreak, as we've had many, many times over the last several years, including during the COVID-19 pandemic. And this creates tremendous economic pressure on the farmers and the producers of those animals in the first place. So, you have to cull, hundreds of thousands of animals in order to make sure that the outbreaks don't spread. And this isn't something that's uncommon: avian flu, African swine fever, etc. So everything I'm saying is obviously very alarming, and everyone who's in the audience and who's on this panel is going to say, great, let's just stop consuming meat, eggs, and dairy, and we're done. I can stop talking. I've been extremely convincing. This is why I was voted top 25 most influential people by GQ. That's great, right? No, Prabuddha's smiling because that's obviously not true. In fact, by 2050, we're going to need to produce 70% more meat to feed the world's population. And if you look at where that meat demand is coming from, it's being driven primarily in places like India as incomes continue to rise. And this is FAO data, UN Food and Agriculture Organization data, but it's borne out by when we have conversations with animal meat producers and people who set up these factory farms. The idea that we're still in this kind of bucolic phase of production in places like India, where it's a few backyard chickens, is just not true. In India and Southeast Asia, Latin America, etc., industrialization has continued apace. So I've said all of this stuff, and what I am telling you now is that eat chickpeas, not chicken, eat broccoli, not beef, simply hasn't worked. We've known a lot of these problems over the last decades all over the world, right? There have been all of these reports, as I said earlier, the Eat Lancet report, the Intergovernmental Panel on Climate Change report, the Game Changers documentary, if you want to reduce your burden of proof a little bit. And there's a lot of work that's been done to shift people away from eating animal-sourced foods, but our theory of change is a little bit different. It is, let's make products, or let's enable making products that taste the same or better and cost the same or less, so that for producers as well as consumers, it feels like a simple switch and not a sacrifice. In short, let's make something people want. So this kind of gives rise to a transformative suite of technologies that fit into three categories that we call smart proteins as a bucket, or alternative proteins is what the world calls them. These are basically plant-based proteins, or plant-based meat, eggs, and dairy, cultivated meat, that is meat cultivated directly from cells, or fermentation-derived proteins in the center there. And I'll be very quick going through these, but plant-based meat is a category that's absolutely taken off globally. It's not your grandparents' soy nuggets. It is products that are made from plant or crop ingredients, but which are supposed to replicate the sensory and cultural experience of meat. So if you look at the bottom right there, that is certainly not a soy nugget. That looks like a chicken breast or chicken meat in general. I want to highlight a couple of companies in this sphere. So this company is San Francisco-based Eat Just Inc. They make scrambled eggs from moonbeams, and they use vastly less water, land, and emit vastly less CO2 emissions, as I was outlining earlier. They've already sold the equivalent. In fact, that number is probably out of date. They probably sold the equivalent of about 100 million eggs now, and they're scaling across the world. Worth noting that their primary ingredient is moonbeam protein, and the moonbeam has been grown on this subcontinent for over 4,000 years. So we really should be competing for these global supply chains and birthing these companies ourselves in countries like India. The second example is the very famous Beyond Meat, which is public on the New York Stock Exchange. A couple of years ago, they went public in the most successful listing for 2 decades. If we replaced one of the 3 beef burgers that Americans eat every week with a Beyond Beef burger, with a Beyond Burger, it would be the equivalent of taking 12.2 million cars off the road or powering 2.3 million additional homes. So what we're talking about is huge impact. But for the consumer, all it means is I'm eating breakfast or I'm eating lunch and I'm having major impact on the planet, which is wonderful. Just to highlight, as I said earlier with Eat Just Inc., we should be competing for the global supply chain and the global manufacturing capacity in this space. So there are opportunities to optimize across this entire value chain, whether it's from crop optimization to raw material processing, all the way through to end product formulation, distribution, etc. And India should be seeing itself as a cradle of innovation in this space. And all of this is projected to be extremely lucrative over time. It already is. If you look at any of the projections in the space, we're talking about anywhere from $100 billion to $370 billion of projections from the likes of A.T. Kearney, Barclays, J.P. Morgan, etc. I'll dwell very briefly on fermentation because Ori already mentioned what Remelt is doing. But essentially, fermentation is a very, very well-established area of technology, as we all know. And even our ancient ancestors, relatively ancient ancestors that drank beer and ate fermented foods know what fermentation is. But what we're talking about specifically in the alternative protein space is the application of these techniques that have been used for amino acids and kombucha and all of these things over time to the alternative protein sphere. So there are a few opportunities to do this. There's what's known as traditional fermentation, which, as I said earlier, makes alcoholic beverages, tempeh, etc. There is sorry, yeah.
Yeah, maybe in a minute, if that's okay.
Yeah, I'll wrap up in a minute. Yeah, so there's whole biomass fermentation, and there's precision fermentation, which is what Ori and his team are doing. Just a couple of examples of the products here. I mean, these are truly transformative opportunities to create products that taste the same or better and cost the same or less. And then lastly, cultivated meat. I'll round out right after this. This is taking a biotech process of cell culture and applying a food lens to it to scale up and to provide what is biologically identical to meat, but cutting out the middle animal. You don't need to cycle these calories, these nutrients through the animal. You farm the cells directly instead of farming the animal and slaughtering it. And this is what the value chain for that looks like as well. So I'll stop here. The entire point of this presentation was to say we're having our horizons broaden, just as Melanie said earlier. We're scanning the horizon. We're seeing a lot of great technologies that have truly transformative potential in terms of human and planetary health. And we'd be glad to work with people in the audience and on the panel to take this forward in the country.
Thank you so much, Varun. That was fascinating. I'm sure that in some other panel, we would definitely be meeting up and we want to hear more. This was fantastic. So delighted to hear you. Right now, we have our next speaker, Dr. Sarvan Sarkar, who is a Birmingham Fellow in the Institute of Cancer and Genomic Sciences in the University of Birmingham. He will be speaking to us about harnessing human stem cell-based platforms for therapeutic exploitation of autophagy in human diseases. Dr. Sarkar is a Birmingham Fellow at the University of Birmingham and holds the distinction of being a former Fellow for Life at Hughes Hall, University of Cambridge. He did his PhD and postdoctoral research at the University of Cambridge at the UK, doctoral research at the Whitehead Institute of Biomedical Research at MIT. Dr. Sarkar studies the biological process of autophagy, to which we are going to look intently forward to. So, Dr. Sarkar, over to you.
Thank you, Prabodha, for the kind introduction, and thanks to all the organizers. Yep. So I'm here. Can you hear me?
Yeah, please.
All right. Yeah. So thank you, Prabodha,
Yes,
for the kind introduction, and thanks to the organizers for inviting me. So I am a basic biomedical scientist, and so I will be sharing our research today. And can you see my slides? Yep. So I'll be talking about how we are using human stem cells to study basic biological process. In this case, I'll be talking about a process called autophagy and how it can be used for understanding disease mechanisms and drug discovery. So a brief background about human pluripotent stem cells. They comprise of human embryonic stem cells, which can be found in the inner cell mass of a blastocyst of a human embryo, and as well as human-induced pluripotent stem cells, which can be derived from a healthy individual or patients, such as from their skin cells, which can then be reprogrammed into iPS cells. And these human pluripotent stem cells have the ability to undergo self-renewal. That means they can propagate themselves, as well as they are pluripotent, means that they can be differentiated into different cell types of the body, and this enables us to create relevant experimental platforms to undertake human relevant biology. In our lab, we are using human ES cells and iPS cells. This is some pictures generated in our lab from which we are making different cell types such as neural precursors and neurons, as well as immune cells like macrophages and also fibroblasts. And the whole idea is to create an isogenic cellular platforms which all have the same genetic background so that we can study cell type specific effects. And our main interest is understanding the basic science such as the role and regulation of autophagy in the human system, as well as drug discovery of autophagy modulators that have important biomedical applications. So a brief background on this human pluripotent stem cell platform and why it is important, because we can now derive patients' fibroblasts and blood cells, such as let's say in the context of neurodegeneration, a patient with neurodegenerative disease. And these cells can then be reprogrammed into iPS cells and then further differentiated into brain cells like neurons, which are the relevant cell types affected in the disease, which are otherwise not accessible directly from the human brain. So we can really create an experimental platform where we are interested in disease modeling and drug discovery. So the biological process we are primarily interested in is called autophagy, which is an intracellular degradation pathway. For example, it can clear unwanted and undesirable materials inside the cell, such as damaged organelles and aggregated proteins, and they're all degraded inside a vesicle called autolysosomes. And by doing this, autophagy provides a housekeeping function, like the clearance of undesirable materials from the cells, and the breakdown products actually goes into various metabolic pathways to provide energy and metabolic homeostasis. Now, this function of autophagy is very critical for cellular survival as well as human health, and therefore, autophagy has been implicated in multiple human physiological and pathological conditions, ranging from aging to cancer to infection to myopathies, liver disease, as well as in neurodegenerative disorders. Now, in my talk, I will be focusing on the role of autophagy in neurodegeneration, and its role is primarily linked to the fact that this process can degrade the mutant toxic proteins associated with neurodegeneration, as well as it can enhance the clearance of, let's say, damaged mitochondria, and this clearance of these undesirable materials from human neurons is very important for their survival. So we have also used our human stem cell model to create an autophagy knockout system in order to show genetic confirmation that how important is autophagy in human neurons. So we can differentiate this autophagy deficient human ES cells into neural precursors and further into neurons. And you can see in all the autophagy knockout cell types in ES cells or neural precursors or neurons, they have increased cell death. And this actually suggests that basal autophagy is critical for neuronal survival, and autophagy malfunction, which is reported in several neurodegenerative disease as well as it happens during aging, contributes to neuronal cell death or neurodegeneration. Now, in the past, we have identified a number of autophagy regulating pathways, and I'm not going into the details, but here are some of the key autophagy inducers that can enhance autophagy, and these have been shown by many labs that they can rescue disease phenotypes in transgenic disease models. So in a way, it is proven in many disease scenarios that stimulation of autophagy could be a therapeutic strategy. Now, on the contrary, autophagy dysfunction has been reported in multiple neurodegenerative disease, ranging from Alzheimer's to Parkinson's to Huntington's disease and so on. And as well as it's also reported in certain other early onset, like childhood onset neurodegenerative conditions, as well as lysosomal storage disorders. So here we are trying to establish defective autophagy as a shared mechanism so that we can target multiple rare disease having a same disease mechanism. And in this case, we are today I will talk about Wolfram syndrome and Niemann-Pick. These are just highlighting two of the rare diseases we are studying where we have established defective autophagy. And our ultimate goal is to target autophagy as a therapeutic pathway so that it can facilitate cluster trials in these rare diseases in future in association with Birmingham Center for Rare Diseases. Now, previously, we generated neurons and hepatic cells, the two disease-affected cell types, from Niemann-Pick patients. Okay, and in these cells, we see that the patient-derived cells have more basal cell death compared to the control cells, be it neurons, be it hepatic cells. And I'm not going into the details here, but the data show that there is impairment in autophagy, such as accumulation of this autophagy substrate called P62, which means that autophagic flux is defective in these cells. Now, of biomedical relevance, we found that treating these patient cells, disease-affected cells, such as neurons, or hepatic cells with autophagy inducers such as rapamycin or carbamazepine can rescue cytotoxicity in patient cells. And we see some other more autophagy inducers such as trehalose and verapamil were cytoprotective in patient neurons. Now, what is interesting, we found that there is cell type specific mode of action of autophagy inducers, such as some drugs that are working in neurons may not work in hepatocytes. And in addition, drugs that are identified in immortalized cancerous cells like HeLa cells may not be effective in patient-derived disease-affected neurons. So therefore, it really shows a need for actually undertaking these studies in relevant human cellular platforms. And of course, our study here suggests that autophagy induction is cytoprotective in vitro NPC1 patient-derived neurons. And similarly, we have been using recently patient-derived neurons from Wolfram syndrome. It's another rare childhood onset neurodegenerative condition where we characterize defective autophagy. And indeed, the neurons derived from Wolfram patients have more cytotoxicity, means that there is more neuronal cell death in patient neurons compared to the control healthy individual derived neurons. And again, we used the same drugs that were effective in both the affected cell types of Niemann-Pick, rapamycin and carbamazepine, and we found that these drugs were also cytoprotective in Wolfram patient-derived neurons. And therefore, our data suggests that autophagy induction is also cytoprotective in this condition. So overall, I have kind of briefly highlighted two rare diseases, Wolfram syndrome and Niemann-Pick disease, where we have characterized autophagy defects, which I have not presented in this talk. But more importantly, we have shown that therapeutic application of autophagy inducers could rescue these autophagy defects, and it can improve cell viability. And not only that, it can rescue many other disease-relevant phenotypes, such as mitochondrial dysfunction, ER stress, cholesterol accumulation, and so on. And
Dr. Sarkar, I would appreciate we're running. Would you be able to sum up in a minute, please?
Yes, absolutely. So the ultimate pipeline is to translate discoveries made in genetic models to early onset rare neurodegenerative diseases, which can then be translated hopefully into late onset neurodegenerative diseases like Huntington's and Parkinson's, and we hope to enable clinical translation in the future. But the important question is, we need to identify the most important autophagy inducer for this clinical translation. And for this, we have generated an autophagy reporter human ES platform from which we can make various relevant cell types, such as the brain cells like neurons, where we are currently doing high throughput image-based chemical screening to identify autophagy inducers relevant for neurodegeneration. And additionally, we are also doing the similar approach in macrophages, which are immune cells, and this could be relevant for infectious diseases like tuberculosis. So I'd like to thank all my lab members and key collaborators, and more importantly, the funding agencies. Like we have recently got, in the recent past, we had got funding from UCARI as well as Newton Bhabha. And I just like to highlight India Institute at the University of Birmingham, which also provides India Institute Visiting Fellowship to foster collaboration between Birmingham and India. Thank you very much.
Thank you very much, Dr. Sarkar. Now we move to Dr. Kapil Khullar. So I would like to welcome Dr. Kapil Khullar. Dr. Kapil Khullar is presently working as a Senior Director in Thermo Fisher Scientific India. He is responsible for the chromatography and mass spectrometry business in India and South Asia. He's also a senior member of the Board of Directors of Thermo Fisher Scientific India. He's been with the company for almost 15 years. Dr. Khullar did his PhD in analytical chemistry from IIT Delhi, and he has been a past vice president of the Indian Association of Analytical Industry from 2009 to 2012. Dr. Khullar, over to you, sir.
Thank you, Dr. Prabuddha and Dr. Sai, for this opportunity. And also, there was a technical glitch, so my computer crashed in the last minute. So thank you for swapping this presentation. I'm not keeping the video on so that I don't end up having challenge in the data transfer. Okay, so I see very interesting presentations on the biotechnology arena, and this is a slightly different subject, analytical technology, but it is important in all the field of biotechnology, whether it is agriculture biotechnology, or it is food biotechnology or pharma. Today, in the interest of time, I'm restricting myself mainly on the pharma or biologics, R&D and QA/QC. Before I start the subject, I will briefly talk about the company for which I work very briefly, and also the context of how the biopharma market is emerging. So very quickly, Thermo Fisher has 80,000 plus employees worldwide, with more than 5,000 employees in R&D and engineers, and we invest close to $1 billion every year in R&D, and that's why we see every year new technology emerging from Thermo Fisher with a revenue of little over 30 billion. So this is just talk about the scale. So with this scale, we are able to provide unique customer value proposition, technology innovation, leadership, and unparalleled global reach. But most important for us is to enable our customers to make world healthier, cleaner, and safer. That's the mission which every employee of company lives. Just to talk about the mission, we were in the forefront of fight against COVID, against the pandemic, both in terms of providing the testing kits and also our electron microscope were used to identify the structure of the virus. So more than almost 50% of the diagnostic kits worldwide were supplied, as well as in India by Thermo Fisher Scientific, and we feel proud of that. So coming to the global biopharmaceutical market, so if you look by molecules, from 2017 to 2025 scenario, the most of the research is on MABs, monoclonal antibodies. Have interferons, insulin. That's a big chunk of research or production happens on that. Growth and population factors, vaccines, hormones. So I think the scenario would remain more or less similar till 2025. That's the forecast period. So monoclonal antibodies segment held a dominant position since 2017 and will continue. So if you look at what are the driving factors, the large number of the recent or upcoming patents which are expiring. This is a snapshot of how many patents have expired and the list of patents or biological drugs which will be off patent, and that's the key driving factor. Many companies, especially in the emerging economies, are developing biosimilar. Of course, the innovator drugs may not reach to the masses or may not be affordable. So I think the solution is to develop biosimilar which are more economical. And this slide talk about, it's also very time consuming to develop a new product as well as the investment. So if you look at the biosimilar piece and compare that with the innovator, it's around $100 million to $200 million kind of investment compared to the innovator drug, and that's how it becomes more affordable. So that's just to set the context. So now talking about the challenges in the biopharmaceutical industry, analytical testing specifically, both during the development stage or production stage. We need to find out number of parameters, whether it is higher order structure of proteins, the primary structure, protein variation, impurities, to find out routine QA/QC or bioanalysis and DMPK analysis. So there are a number of technologies both in the separation science, whether it is chromatography like BioLC or ion chromatography or the liquid chromatography columns, or in the characterization, which is on the mass spec. So in the interest of time, I will spend more time on the mass spec, which has revolutionized the protein characterization. So this slide presents the various workflows which are essential in biopharma. The challenge in the past was that we were not having the powerful mass spectrometers which can look deep inside into the proteins. And I will also highlight here the notion which was there on the mass spectrometry, that mass spectrometry cannot go beyond 50,000 mass resolution. So it is only a technology for the small molecules or generic pharma. But with the invent of Orbitrap platform, not only it passed 50,000, it breached the barrier, psychological or scientific barrier of 100,000. And today we have very, very powerful mass spectrometers which can go to 500,000 or even 1 million resolution. So not only we can look deep into proteins, but we can also have the structure, cell biology, or the structure or cancer cells identification or characterization. Now, so this is a data. I'm not going more into scientific detail, but this data shows how much is the improvement in terms of the quality of data compared to the previous mass spec and with the invent of Orbitrap. And this is the data which is published recently in 2017, which differentiate. It has come in the Analytical Chemistry Journal, which talks about the quality of data and one more important thing is that this is the data which now the FDA is asking the quality of data, so that the innovator and the biosimilars can be compared in terms of its efficacy and performance, and also batch-to-batch production. So this is about the development stage where you need mass spec for various workflows. So all in all, there are almost 25 parameters which are to be monitored during the development stage, and also during the production stage. The current conventional methods, there are a number of methods. The biggest challenge is not only development, but how do we have consistent batch-to-batch production? So this slide presents, the attributes which has to be measured and various techniques to be used. Now, the biggest challenge for the scientists is hugely time-consuming. Can we work on a method which is fast, which is efficient? So the current trend is working using HRMS, and teams globally are working, the global biopharma companies and Indian biopharma companies on multi-attribute method. So which will help both during development stage, especially in the QA/QC. So this is the emerging trend that we can measure. Currently we are in a position to measure almost more than 12 parameters together. But I think the work is in terms of developing more workflows, so that both at development stage as well as production stage, the analytical technologies make life easier for biotechnologists. So that was my idea of presenting the analytical emerging analytical technology. And the last slide is with the power of mass spec, we can now measure the entire virus mass. This is an example where a virus mass of 9.3 megadalton, whole mass has been measured using the advanced mass spectrometry. So with that, I hand it over back, Dr. Prabhuda, to you, and we'll be happy to take questions at the end of the presentations.
Thank you so much. I think you really helped us with this talk. Thank you so much. I quickly call upon Mr. Swapan Mehra, who's the Vice President of Invest India. Swapan is the Vice President at the Waste to Wealth Mission of the Office of the Principal Scientific Advisor to the Government of India and Invest India. Swapan is an environmental technology and finance expert with deep expertise in developing multidisciplinary solutions for bringing together finance, policy, ecological economics, ICT, and clean technologies. He serves on several boards. So with that, if I could request Mr. Swapan to just come on to start with his talk, please. Welcome to the panel.
Thank you very much, Mr. Kundu. And I'd like to start off by saying that I've been observing other speakers, and what I work on and what I'm about to say is extremely different from what is being discussed here. So I don't know whether that is good or bad, maybe refreshing for all of you, or maybe an opportunity to take a break. I had decided that I'm just going to talk, but observing the trend here, if you don't mind, I'm just going to share my screen and take you through 2 slides, just 2 or 3 slides about my current work and the mission that I work on. Just a second, please. Sorry. Yeah. And then I'll just finish in 5 minutes, and then it'll be great to discuss. So I represent a mission called the Waste to Wealth Mission. I now work at the Office of the Principal Scientific Advisor to the Government of India, and my mission is one of the 9 missions of the Prime Minister's Science, Technology, Innovation Advisory Council. Now, the goal of this mission is to provide science and technology inputs to identify, test, validate, and then scale up technologies that address India's waste challenges across air, water, land. So we have broad mandate. We do this through a 5-pronged approach. We identify key critical waste problems, potential solutions in the form of national, international technologies that need to be adapted, customized, updated. Then we demonstrate these in locations and try and create exemplar pilots by leveraging resources from the various government ministries, institutions, states, as well as through a number of international tie-ups. We are a 6-month-old mission, and we are about to implement a 10th project. So it's early days, but it's been rapidly getting very active for us with a lot of interest from various partners. And then we focus on promoting scale-up of chosen technologies through the horizontal access that the Office of the PSA gives us across the government, but also in partnership with various other actors. We work a lot with industry associations, with funders, so on and so forth. We are in the process of creating a national portal called the Waste to Wealth portal, which is expected to become the de facto portal of technologies on the waste challenges of India. These are some of the technology areas that we are currently working on. We are working on cleaning water bodies, sewage treatment, agri-residue management. These are pilots which are already underway or implemented on cleaning water bodies in Delhi, in Dal Lake, in Jammu Kashmir, on innovative technologies that use plastic and turn plastic into either liquid fuel or are able to recycle plastic, so on and so forth. So we identify technologies through challenges jointly with various partners, or we also invite technologies to submit their interest on our portal, and then we work with them to create solutions and then deploy them. We have a panel, an expert panel, which we are populating very rapidly. We've got a number of experts from Indian academia, but also industry, who help us validate monitoring data from each one of these and conclude whether these technologies fulfill the Indian context in terms of both technical applicability, but also economic financial applicability. I'm going to stop here. I'm going to conclude my presentation. And then I'll very much like to listen to all of you if you have any questions or feedback. Like I said, I think that this is a very good digression for all of you, but an important issue nonetheless, and would be very interesting to discuss overlaps with your work, if any. Thank you so much.
Thank you so much, Sapan. I mean, that was fantastic. I mean, I'm fascinated. I may have a couple of questions myself for you, but I will leave it to the audience and other panel members to take it on. Thank you so much. With that, we are ahead of time, and that's fantastic. I really thank everyone for your cooperation. It has been phenomenal listening to each and one of you. So, Dr. Sai, is it okay if we jump straight away into questions for each of the panelists? Is that okay?
Right. Yes, sir. Perfect.
Fantastic. Professor Melanie, there's a question that came through is, in the post-COVID-19 world, since you are in charge of policy and how funding takes place, and you are in a position to sort of direct research, the question is, how will your organization respond, or will it change its approach in a post-COVID-19 world to the way it seems? Sorry for putting you on the spot, but I thought it was a relevant.
So I think it's a good question. I think we've been adapting throughout the pandemic, actually, and boosting the research that we've been supporting to find solutions to tackle the pandemic. And I think we can all celebrate the achievements of science, actually, in helping us find routes out through really rapid vaccine production and rollout. But I think it's actually taking inspiration actually from a lot of the topics that we've covered here and thinking about, we have had the most unusual year. We know what some of the really big challenges are, particularly around the climate resilience, food production, human health. I think we really need to focus on some of those things and really bring together, a real sort of cohesive effort to tackle some of those challenges, to really focus on some of those really key aspects. I think we've demonstrated that we can do global collaboration really well through the pandemic. I think we can use that as a platform and do that better. I think certainly for BBSRC, the whole space around biorenewables, bio-based, nature-inspired solutions, I think is really, really key. And we mustn't forget about the fundamental underpinning either. But I think now is the moment to think differently, actually, and to really work together.
Thank you so much. Nadav, a question came in, and you can take either or, is you spoke about type 2. Are you focused on type 1? And B, you talked about vaccines. The second question was, if at all, is antibodies on your spectrum?
Okay, so regarding type 1, yes, the answer is we are definitely working on type 1 as well. With the type 2, currently the phase 3 is being conducted on the type 2, which make about 90-95% of the total diabetic population. But type 1 will benefit tremendously from the oral insulin. Regarding the pipeline, antibodies, and so on and so forth, we obviously, as a public company, once we have more material and concrete information, we'll definitely share it, but we're definitely on the outlook for relevant opportunities all the time.
Thank you so much. Ori, there is two parts of that question to you. And Ori, are you there, Dr. Kuari?
Yeah, I'm here.
Okay. Yeah, one is, what do you believe will be your price to market is one question, and will the nutritional value, because a lot of the world focuses on nutritional value of milk or considers milk as a key protein, especially vegetarians and so on and so forth. So these are the two questions for you: price to market and the nutritional value.
Two very good questions. So the price is, I think it's the main issue here. Our goal is to achieve cost parity at first, and even go lower than the current prices. This is a major challenge. You have to have a well-optimized system to do that. We believe that we have the way to do this, and we have initial indications that we are definitely on the track to do this in actually the very foreseeable future. But it does give you, I mean, having this alternative way, this non-animal protein and non-animal based products give you some kind of an advantage where you can actually go into market even if the price is still a little bit above, let's say, cost parity in this type of product. So to answer this question, I think we have the opportunity to go into market before we are at cost parity, and then achieve and go below cost parity as we move forward. Regarding the nutritional values, to us, the nutritional values will be similar and better than the nutritional values of animal-based products. They will be much safer, much more controlled. You have a full control of what to put in. For example, lactose, which 60% of the world population is lactose intolerant, will not be a part of this product. The types of fats and sugars and the amounts that you put in, of course, will be tightly controlled. In the bacteriological sense, in having the control, not having antibiotics inside a product, and also other aspects. So we think that our product will be safer and more healthy for human consumption. And of course, the protein is the same, so you get this very optimized amino acid composition that is found in animal milk.
Thank you very much. At least people like me who are severely lactose intolerant may generate a lot of benefit. Fantastic. So coming to Varun. Varun, two questions again for you that's come through is, one is, the proteins that you're talking about taste to Indians, and do really investment in such technologies, would it be beneficial for a country like India?
Yeah, thank you for the question. So, I mean, it's important to note that much of the growth in animal protein consumption, a significant portion of that will come in places like India, as I mentioned. So, you know, these things have been, this is a result that's been proven over and over across the world. As incomes rise, people want to consume more animal protein, right? So, as an example, I'm 3 kilometers away from the largest slum in Asia. And if we go there and we talk to a family of 4, they will say, look, we're eating 1 mutton meal a week for 460 rupees. And if we could, we would eat 2 for 920 rupees. We'd eat 3 for 1,380 rupees, and so on. So what we're saying is we'd love to be able to provide at every level of the socioeconomic pyramid foods that people know and love and that fit into their heritage, but without the same implications on land use, water use, greenhouse gas emissions, antimicrobial resistance, etc. And, you know, just thinking about this for a second, we talked about waste to wealth, right, with Swapan's excellent talk. If we're talking about 9 calories into a chicken to give you 1 calorie out, that's 800% waste. We should think about that as waste. And chickens are the most efficient animal. Cows are much, much worse. Pigs are worse than that also. So, I mean, look, there's the question of nutrient density, there's the question of all of these things. And in order to match the taste and the nutrient profile, we do have to make investments. As you said, we do have to invest in R&D and commercialization and all of those things. But this is the critical decade for us to steward planetary health. And a lot of those things will be decided in places like India.
Thank you so much. Dr. Sarkar, there was a question, there's a couple of questions obviously came in because of the scientific content of your talk. But one was a question, a very simple question that came through and I sort of struck my eye. Is there any relationship with autophagy and cancer?
Yes, so there is a complex relation. It's kind of it's called a double-edged sword. So it's at kind of different stages of cancer. Autophagy can play, differential role. For example, when tumor is forming and they are deprived of nutrients, so that's when they trigger autophagy to survive. So you might want to inhibit autophagy to kill the tumor cells. And indeed, there are, there has been some clinical trials like with hydroxychloroquine, for example, and there are some, reviews in Nature Reviews Cancer. And on the other hand, when somebody is undergoing chemotherapy, for example, you want to, there is a concept, if you trigger excessive autophagy, that can also be, in conjunction with the chemotherapy. So yeah, the relationship is complex, and whereas in other disease scenarios like neurodegeneration or infection, the relationship is more, singular, like in a linear fashion.
Thank you. Thank you so much. Dr. Shashem, any further questions coming in? Let me look at them. So Dr. Kullar, thank you so much. I don't seem to have too many questions that have come your way, unfortunately. I may have one coming in for Dr. Sapan, which is coming in. Give me a second, please. Dr. Shashem, any further questions for any of the speakers, please? Okay, then if it's okay, I can ask Sapan a question from my side, if that's allowed. Is that allowed? Can I ask one from my side?
Please, please.
Yeah. So it was fascinating to hear the kind of, cleanup job, waste to wealth that you're doing. If you would like to take from the list that you showed, would you like to just take us through maybe one of the projects? We have maybe a minute or minute and a half, and just speak about one key project that you're doing as an example. Would that be possible?
Sure, absolutely. I'd like to, for example, talk about the paddy crop residue work that we are doing, for which we've just implemented a pilot. We've implemented a torrefaction unit in partnership with the National Agriculture and Botany Institute, NABI, in Mohali. This was implemented in Mohali because we wanted a site close to the paddy residue. This torrefaction unit, which at the moment torrefies only some 20 to 30 tons per hour, has proven that with the right technology, you can enhance the calorific value of the paddy residue substantially. And now there is great interest from our power companies to see if we can scale up this torrefaction, also try and come up with similar solutions at a lower scale and cost, so that we can install these across the states of Haryana and Punjab and be able to convert a lot of the residue into fuel. Now, when we started discussing this, we realized that this can be done. However, the big challenge there is that you do not have infrastructure for collecting waste, because you need to be able to, as we all know, that we need to be able to clean the fields in a very short period of time between the two crops, between paddy and wheat. We realized that there is a big problem because the mechanization required is extremely large, and at the moment resources are very little. So there is one startup which was founded by a farmer and his family, which is a leader in this space, who clean about 50,000 hectares of land every year and are able to collect 100,000 tons of paddy residue. Now, this is the largest, and the challenge that we have is that we have to multiply this by 170 to be able to achieve the whole scale. And we are now looking at launching a challenge very soon, in which we are inviting consortia to apply, where they can combine and evolve better sharing models of these mechanical devices, innovate at low cost devices, and also be then able to combine them and have a bouquet of uses, such as we can optimize on returns, because we realize that things like furniture, tableware give a much higher return but lower quantity. Energy uses a higher quantity, less price, right? So this is some of the work that we are trying to do in this space. Similarly, in East Delhi, we have a project in which we are trying to clean urban drains. Many of you who have been to Delhi or East Delhi would have seen these urban drains, which are completely clogged with construction demolition waste, plastic waste, and also by the way, biomedical waste. When we clean these drains, we have mechanical removers. We find a lot of syringes and needles and blood bags in them, which is really scary, right? So we have put in place 2 machines which mechanically remove the solid waste, and now we are looking at using that waste through in a partnership with Indian Institute of Plasma Research by putting up smaller plasma pyrolysis plants which are able to generate heat out of these, and then be able to use that heat in situ within our technology park for some other purpose, and have low-cost sewage treatment plants, decentralized plants, which can take this water and start cleaning it and making it of some acceptable quality. So these are a couple of projects that we are working on, and all very challenging, all very exciting. Yeah, thank you.
Thank you. I think we have maybe time for just one more question, which I think Dr. Sarkar, yes, you put up your hand. So one question, please, if that's okay, because I think we're right at the end. And I think Dr. Kashyap will also need some time. Yeah.
Yes, so I have a question for Ori and also to Varun. Outstanding talk, fantastic work you guys are doing. I'm super impressed. So I'm thinking from a societal point of view. I mean, although you have given a lot of environmental numbers, I'm thinking that if you make smart food, a lot of industries will be wiped out, especially, let's say, dairy industry. Especially in a country like India, there will be a lot of loss of jobs, for example. So have you, thought of some numbers or addressing the societal problems about, people getting, robbed of, jobs or something for, let's say, poor farmers? Of course, this is nothing to, this is just a societal thing, and what you're doing is far more important about the planet and the environment.
Ori, if I may just address the dairy, the Indian part. So I think these are equally important, right? Because if we're looking at shepherding in a future, no pun intended, I guess, with the word shepherding, but if we're looking at shepherding in the future, we need to make sure that everyone's a part of that future and has a voice in that future, right? So what we're talking about is definitely going to be a diversification. What we're talking about is going to be a transition. Even the most advanced countries in our space, like the Netherlands, are seeing this, they're literally calling it the protein transition. So while some of these technologies are harbingers or indicators of what's already happening in society, right? Consumers are demanding to move away from some of these animal-sourced foods. And so you're seeing dairy companies in places like the US go bankrupt. That is a consumer side of it. What we are saying is we would like to be able to offer an alternative that even gets producers from some of these legacy industries into the future of food. So as an example, we are working in India with smallholder farmers who might be doing aquaculture in shrimp, and we're helping them also do aquaculture in seaweed. And seaweed can be used as an ingredient into alternative seafood applications. It already has all of the, on a molecular level, on a biochemical level, it has phospholipids, certain flavor compounds, etc., that lend themselves really well to plant-based fish as an ingredient. So these are the kinds of things that we're doing. We're very cognizant of the socioeconomic implications, and we're certainly empathetic to that. So that's not our intention or the way things are going. And again, it's worth noting that we need to build in resilience into agri supply right now. A lot of what we do, if we do nothing, a lot of what will happen will be the destruction of those industries anyway, because of the nature of the way the world is going. So as I say, farmers are not going to be adequately compensated when their entire stock of chickens and their entire stock of pigs dies. There is not an adequate compensation. They've taken weeks, in some cases years, to grow that stock. And so we're trying to build in the resilience as well. So I think we're all a part of this, and it's going to be a gradual process.
Fantastic. Thank you so much. Thank you. Ori, would you, maybe 15 seconds, I know all of you are very busy and you may have to jump off. Would you like to respond, or can we close the session?
I agree. I completely agree with Varun. I just said, generally speaking, human beings have had these concerns for a long time, ever since the Industrial Revolution, basically, and automation and technology have always had the problem of replacing human labor. But when you look at the picture and what has actually happened is that the overall wealth of human and the standard of living increases, right? So everyone, at the end, it's not like people were left without a job and the poverty went up, but on the contrary, poverty goes down as technology moves forward.
All right. Thank you so much, esteemed panel members. I'm really, really grateful to you all, delighted with the amazing talks we've had. So thank you so much. Dr. Shashem, over to you.
Thank you, Dr. Prabuddha. And I think I should take this opportunity to thank each one of you, each one of our esteemed speakers on behalf of BioRack. I think each of the topics that we had were covered extensively, and we had enough time to discuss. And thank you for staying throughout the session and then taking part in the discussion as well. And I thank all of you who have joined this session on behalf of the organizers, and would request you to actually join in the closing ceremony. Thank you. Namaste and Jai Hind. Bye.
Thank you. Thank you, everyone. Thank you.
Thank you.